Good afternoon to the first session of day 2, e-FDP and SDP, organized by Cochlear Education Societies, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, in association with Indian Pharmaceutical Association, Nasik branch, on evolution in teaching, guiding value in learning and social understanding through research. I welcome Dr. Dinesh Dhumal, resource person of first session of day 2 of 5-day FDP. I welcome moderator of the session, Professor Hemant Chikle. I welcome coordinator, Dr. Rupali Patil Madam, Dr. Shilpa Harak Madam, on this first session of the day. First of all, I would like to introduce Professor Hemant. He is assistant professor in pharmaceutical chemistry at Cochlear Education Society, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, since 5 years. We have young and dynamic speaker today, Dr. Dinesh. I request moderator of the session, Professor Hemant, to introduce Dr. Dinesh. Please, over to Professor Hemant. Please, sir.
Thank you, sir, for your kind words. Now, yesterday we have a very good session of both the dynamic speaker. So today, again, we are having Dr. Dinesh Dhumal, sir, who is working as a CNRS Post Doctoral Researcher at Center Interdisciplinaire, the Nanoscience, the Marseille, France. Qualification and experience. Dr. Dhumal, sir, has completed his PhD in pharma technology from Institute of Chemical Technology, Mumbai in 2018. Sir did his post-graduation in pharmaceutics from North Maharashtra University, Jalgaon, Maharashtra, 2011. Sir completed his graduation from North Maharashtra University, Jalgaon, Maharashtra, 2009. As a part of his PhD, Sir is a Raman Charpak Fellow at Axis Marseille University, Marseille, France, in 2017. As far as the awards and recognition are considered, the Sir received Best Oral Presentation at ACNRS 2020, Chennai, India. Sir is a recipient of International Travel Grant in 2016 by DST SERV. Sir is also a recipient of CICS Travel Fellowship in 2016 by the Indian National Science Academy, India. Sir is also a recipient of Biotechnology Leadership Camp, that is BioCamp, in 2006 by Novartis. Sir is the second winner in Best India 2014 by ABLE and DBT India. Again, Sir is a recipient of third winner in International Annual Conference 2014 SACA-CCP, Mumbai, India. Sir is a DST Inspire PhD Fellowship in 2012 to 2018. Sir is a gold medal rank holder in Master of Pharmacy in North Maharashtra University, Jalgaon. Sir is also a recipient of Sir Ratan Tata Trust Academic Scholarship in 2010. As far as the research outputs are considered, the Sir interests include interdisciplinary field like functional dendrimers, biomaterial or biomedical applications, nanomedicine, that is dendrimer micelles, liposomes and nanoemulsions, then targeted or non-targeted for antimicrobial and anti-cancer drug delivery. Then Sir is also having a keen interest in gene therapy also in analytical method development, that is stability and impurity profiling. Rather than this, Sir is also having a one patent to his credit and one international book chapter to his credit. By this particular research output, Sir has citation of his reputed publication about 116. He has H index of 6 and I-10 index of 4. So, on behalf of Gokhale Education Societies, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I welcome you, Sir, on this e-FDP and e-SDP. The title of today's talk for Dr. Dhumal is Amphiphilic Dendrimer for Biomedical Application. Now I will share this e-platform to Dr. Dhumal sir to kindly enlighten with this topic. Over to you, sir.
Thank you so much for your kind introduction. I did my master's in pharmaceutical chemistry, not in pharmaceutics. Okay, I'm sharing my screen now. Is it? Can you see my screen? Is it there?
Yes,
Pingade
yes,
sir?
yes, 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 Dr. Dinesh, proceed,
Hello?
please. Yes,
Okay,
yes,
okay.
yes.
Okay, thank you. So today I'm going to talk about the amphiphilic dendrimers and their biomedical applications. And here during my postdocs, now I'm working on amphiphilic dendrimer, and I would like to introduce work of my research group, what we are working here. So just to start, like we know, we all humans suffers from many health diseases. They are inherent acquired diseases, emerging pandemic infection, like we currently suffering from COVID now. It's a disaster for us. Then there are aging population and age-related diseases. And to overcome these diseases, the drug discovery process in the pharmaceutical industry is going on. But as we all know, for a one drug approval, it takes minimum 15 years. We start from 10,000 to 20,000 compounds, and we come up to a one molecule only at the end of spending too much money. And despite of that, whatever the marketed or FDA approved molecules are there, most of them, more than 40%, are mostly water insoluble, or they have a very low drug ability. So these are the issues related with this drug. Already we suffer from a very long process, put so much money, so much time, so much manpower. At the end we come up with the molecules, but again this molecule has a problem, like a non-soluble, very low drug ability. And most of these molecule fails in preclinical also because of their physiochemical characteristic issues and all. So to overcome these issues, then the nanotechnology based drug delivery systems has a promise, and it gives us opportunity to improve that process and to have a more better results. So what we can achieve through a nanotechnology, we can improve the solubility, we can reduce the toxicity, we can control the drug release, which is necessary for in certain diseases. Then we can improve bioavailability of this drug. Then later we can have an active targeting. So to achieve all these things through the nanotechnology, we have a different drug formulation, nanoformulation, drug delivery systems, and we have different nanocarriers like lipids, there are polymers, dendrimers, there are metal nanoparticles are there. So, and we formulate them in different drug delivery systems like nanocrystals, liposomes, micelles, MEDs, NEDs, dendrimer, carbon nanotubes. So these are all processes are there. And through this, currently there are more than 1100 nanopharmaceutical products are in development, as well as more than 50 nanodrugs are in the market, like Toxil, Opentero, and like Ambisome for the antifungal amphotericin B. So there are many examples. So from this, we can say that the nanotechnology certainly help us to improve the drug discovery process. But we believe that apart from whatever I said, lipid, polymers, metal nanoparticles, carbon nanotubes, we believe that the dendrimers are ideal carrier for the drug delivery system. Why? Because if I have to say like dendrimer, so it's a Greek word. It comes from the dendron and mers, like a tree-like dendritic feature. So dendron means tree and mer means parts. So it is like a tree-like structure having a parts, branches. It has a well-defined structure, unique multivalency, it has a nano size, and these are ideal characteristics for a nanocarrier, for a drug delivery system. So we believe that the dendrimers are an ideal nanocarrier for drug delivery systems, apart from lipids and polymeric material, other polymeric material. So before going for the work we are doing in our group, I would like to introduce some of the concepts related to the dendrimer. So I would like to go first with the conventional dendrimers. So like I would like to introduce dendrimer structure first. So on your screen, you can see that on the left hand side, there is a cartoon displaying the dendrimers, and dendrimer basically has a three parts. First is a central core. You can see the orange color highlighted, where the dendrimer growth begins. Next, it's repetitive branching units, the way I said, more parts, so which allow dendrimer growth geometrically organized in radial layer. So these layers are called as a generation. So you can see in a cartoon like it's G1, G2, G3. And at the end, another final part is a numerous terminals. So like here I highlighted in a pink and violet color. So these are the functionalities which expose to the surface. All these three parts of dendrimers are really essential for their synthesis and their application and their physiochemical characteristic. So based on all these parts, central core, repetitive branching units, and the numerous terminal, dendrimer can be classified based on the what core present, what functionality is present on the surface. So on the right hand side, you can see I have just put a picture of the different types of dendrimer based on their core, like the PPI. So polyamidoamine. So there is that core, so they call it PPI. Then there is a pamam dendrimer. Then there is an ester core inside, so polyester. Then on the surface there are glucose moieties, so they are called as a glycodendrimer. So based on the different functionalities on the surface or the branching units, dendrimer can be classified in different ways. So I would like to say the different biomedical application of the dendrimers, what we can achieve through the dendrimers in the drug delivery system. So like to start from, we can attach a targeting moiety on the surface through partial synthetic way, or we can have them fully attached to the surface. We can tag some moieties. We can conjugate drugs like a prodrug system. We can have enzyme on the surface, electrostatic interaction or hydrogen bonding, or we can directly have a stable bonds with the dendrimer. We can encapsulate hydrophobic drugs or hydrophilic drugs based on the type of dendrimer we are dealing with. We can do the pegylation also. So we have a variety of choices to play with the dendrimer to use them as a useful nanocarrier for different types of drug delivery system. And I would like to talk about the pamam dendrimers, because pamam dendrimers are the most widely explored dendrimer throughout the time. Like since the 1979, the PPI dendrimer started, and then after that in 1982 onwards, the pamam dendrimer has been widely explored. So on your screen, on the left hand side, you can see a chemical structure of third generation pamam dendrimer, which is bearing a triethanolamine core and the polyamidoamine branching unit. That's why it is called as a pamam dendrimer. Then these dendrimers can be synthesized by different synthetic routes like divergent synthesis, conversion, a mix of divergent conversion. But here I just like to share about only the divergent synthesis, because we mostly follow this in our group. And this synthesis pathway is like a stepwise pathway. It is like an iterative step. You have like two reaction sequence. First you need to do Michael addition, and then subsequent amidation until the generation which you want. You want first generation, you have 2 steps. For second generation, 4 steps. So depending on the number of generation you want. And in this divergent synthesis, it is easy to purify as compared to the other steps, as well as it gives you a less impurities. And these help us to achieve the final dendrimer with the minimum impurities, because if you have impurities, it is really problematic for their application, and you might get a false results, and you will not have a desired properties what you actually want in your dendrimer. So this is a classical example I'm giving here for polyamide dendrimers because we working on it. Next, now I would like to start with Dr. Peng's group work on a conventional permanent dendrimer in a nucleic acid therapeutic. So like we have explored this dendrimer for DNA, siRNA, and saRNA delivery. So before going for actual work, we mostly work on siRNA. So I would like to tell a story about like what is siRNA and how it actually works, and what are the problems of siRNA, and why we need a drug delivery system for siRNA, and what should be the ideal characteristic of this drug delivery system. So the siRNA delivery barriers and challenges. The first, what is siRNA? So it's a short interfering RNA. So why this term came into picture? Because Mello and Fire, who won the Nobel Prize for this in 1997 and 1998, achieved a transient gene silencing via sequence-specific mRNA degradation. We all know if we have some disease, there is always, it is associated with the protein regulation, whether up regulation and down regulation. And if we stop that up and down regulation, we can stop the actual start of the disease from its core. So here the up regulation and down regulation of that mRNA can be achieved through siRNA. So we can stop the disease from its roots. So the silencing means the working of mRNA can be stopped by degrading it using a specific siRNA. And this phenomenon using siRNA is called as RNA interference. Means this siRNA interfere with the mRNA properties by cleaving it. So how it works? In the picture, you can see that the siRNA, it then cleave into the single strand by AGOT protein, and then it goes into the RNA induced silencing complex where it attached to the mRNA, and then it degrades the mRNA. So we need a specific siRNA for a specific mRNA to stop the disease progression and to stop the protein upregulation and downregulation. So, but this siRNA, which we want to use as a drug, has a problem in the drug delivery. So what are the problems? So there are different barriers for siRNA delivery. If I have to talk about just the naked siRNA without drug delivery, the naked siRNA suffers three problems. First, it can be easily degraded by serum nucleus. You inject it in your blood, you have a serum nucleus, it will degrade it. So there is no use. It can be aggregated by serum proteins. So that is also disadvantage, and poor tissue permeability. It cannot penetrate the tissues which we want because siRNA is highly negatively charged, and our cell membrane is also negatively charged because of the phospholipid. So they have like opposite interaction, and it's difficult for siRNA to penetrate the cell. So these are the barriers. But if you want to have a carrier system for siRNA, you use a drug delivery system. But still, this drug delivery system suffers major 6 problems. Like there is a non-specific distribution. Next, there is a tissue barriers again, as I said, like if you are targeting for a cancer, then the cancer tissue barriers are there. Then there's a poor cellular uptake. Then inefficient release from the endosomes. Like if you see the process here, I will explain you, like see, the siRNA is there at the first one, then second and third, I explain this. Then in the fourth one, when you have here the non-specific distribution, the body occur. In the fifth, you can see there is a tissue barrier, they cannot enter into the cell. And then if they are come to the cell, there is a poor cell uptake. That is also issues with the drug. Then if they come into the cell, the pathway goes through the endosomal escape. They first go into the endosomes. But here, if they are not able to go escape from the endosome, they will go through out of the cell by endosomal lysosomal cycle. So they need to escape from endosome. And if there is an inefficient release of the endosome, then there will be no adequate therapeutic effect will be achieved, and no proper gene silencing will be achieved. So this is a major problem. Then inefficient release from the carrier. Okay, they release from the endosome, but they should be released from the carrier to go to the targeted mRNA or to get fused into the RCS complex. And sometimes another is off-target effect, which is a minimal side effect, but which is also considered as one of the problems for the siRNA delivery. So these are the problems are associated with the siRNA. So what will be the ideal nanocarrier to overcome the problems of free siRNA delivery? So the drug delivery system should protect because they degrade. The drug delivery system should be non-toxic because else you will have an immune system response. Most of the drug delivery system fail in a clinical trial because of their immune response. So it should be less immunogenic. They should have a renal clearance very slow. If they get eliminated from the body very quickly, then there will be no therapeutic effect will be achieved. The cellular uptake should be high and their endosomal escape should be high. So to summarize, ideal carrier should be sufficiently high charge to bind siRNA. As I said, siRNA is negatively charged, so your carrier or the dendrimer should have a specific positive charge enough to bind to the siRNA. And then the siRNA carrier complex should have sufficient low surface charge, which should show a less immune response also. So they should be less immunogenic, non-toxic. So these are the ideal properties a one carrier should have to deliver a free siRNA for the therapeutic activity. So I would like to share the first work from our group on the conventional dendrimer for nucleic acid delivery of DNA and siRNA. In a cartoon, you can see that our dendrimers are positively charged because the surface is primary amine. They are sufficient charge and they can encapsulate the siRNA with the charge-charge interaction. Then they can have a healthy cellular uptake. They can have the endosomal escape. Later, they come into this cellular cytoplasm and they go into the machinery to break down to degrade the mRNA. And this was a European project, like really long before in 2009, since we started working on the siRNA delivery. So you can see the mechanism how the siRNA interact with our dendrimer complex. This complex later we see it has a particle size less than 50 nanometer. And we check with the gel electrophoresis. As I said, naked siRNA get degraded, but when they complex with our dendrimers, there is no degradation. So the dendrimer protects the siRNA from their degradation from the nucleases. Later, we see the DNA transfection. We can see, you can see on the slide, there is a higher cellular uptake of our G7 dendrimer as compared to the marketed lipofectamine. Later, we check the siRNA gene silencing effect on the protein level, and you can see as compared to the all controls, G7 siRNA shows a degradation of the mRNA on the protein level. So we achieved all the aspects like protection, cellular uptake, and gene silencing. Later, we explored this effective delivery in different RNA therapeutics. Like on your left side, you can see we check on in a liver cancer, where we use saRNA, not siRNA. So saRNA is a activating an RNA, which activate the mRNA, and siRNA is interfering, which stops the RNA. So here we use the saRNA also, and in collaboration with the Imperial College of London. Later, we use similar kind of delivery system in the glioblastoma model and with the Beckman Research Institute in USA, and later we used in ovarian cancer model, and we got a very good result. So later our product, our dendrimer nanosystem with the saRNA, scheduled for a clinical trial in the UK and the USA. But there was a problem. It is delayed due to the GMP production of dendrimers in a large amount. So we solved a problem of siRNA, but later we had a problem with our dendrimer GMP production. So we solve one problem, we have another problem. This is how research works. When you overcome through one problem, you have another problem, and you should have a rational approach to overcome these problems. So the first clinical trial of siRNA therapeutics, which scheduled with the dendrimer, but it got delayed because of the GMP problem. So they didn't use dendrimer, but they used a SMARTicle, the lipid nanoparticles. So although dendrimer was vastly superior than SMARTicles can be produced, but the SMARTicles can be produced in GMP, so have been validated for clinical trials. So here we failed in terms of the GMP production. So we need to overcome this problem. So there is a dendrimer paradox. We are working on dendrimers from last 3 decades, which has a high medical expectation, but there is a very poor clinical translation. That's why you will see a very few dendrimer products in the market, because there is a problem associated with their GMP production and their synthesis. So to overcome these issues, our group started working on self-assembling supramolecular dendrimer. So you can see on the screen, on the left side, there is a structurally flexible dendrimer, which has a molecular weight more than 21,000. And on the right side, you can see an amphiphilic dendrimer, which has a hydrophobic tail and hydrophilic head group, which is a molecular weight nearly to 2,000. And these amphiphilic dendrimer, they self-assemble in a water very quickly. Just you need to dissolve them in water, and they self-assemble. And this self-assembly of this supramolecular amphiphilic dendrimer mimic exactly the structurally flexible dendrimer. So being in a small molecular weight, you can control the synthesis and you can achieve the GMP production. So with the help of these supramolecular dendrimers, we try to achieve, or we try to overcome the problems of the synthesis of the GMP production. But then we need to see whether these amphiphilic dendrimers, supramolecular dendrimer, are as effective as the structurally flexible dendrimer, having low toxicity and good GMP production. So now I will introduce the supramolecular amphiphilic dendrimer work, which is a pioneering work because our group was the first one who used amphiphilic dendrimer for siRNA delivery. So here I would like to just show how we achieved the supramolecular dendrimer in terms of synthesis. We had a hydrophobic tail, which has a azide, and we have a hydrophilic head, which has alkene, and with a simple click chemistry, copper click chemistry. This reaction takes only 2 hours. The purification is really easy, and the yield is more than 80%. So with this, we can achieve the synthesis and the production with a very, very less impurities. And this work of ours was published in Ang1 in 2012. So what are the advantages of this amphiphilic dendrimer over other nanocarriers in clinic? Because as we know, liposomes, many liposomes are there in the market. But what is the problem with this liposome formulation? Most of them are unstable. Take an example of ambisome and fungisome. They were in market, but the problem with them is a batch-to-batch variation and very poor stability. We can go for antibody-related nanocarriers, but they have a very low payload. And for polymer, it's difficult to control structure of polymer, so it is difficult to get desired results with the batch and batch variation. So, but with the help of supramolecular dendrimer nanocarriers, we can control the structure. As I said, we can go from different generation to generation for synthesis. We can achieve a stable formulation very quickly. We don't need any external medium, like no homogenizer, no sonicator, nothing. We have to just put our dendrimer in a water and just vortex for 2 minutes, and it's done. And we have achieved a very high drug payload. The encapsulation efficiency with our dendrimer is more than 40%. So how our supramolecular dendrimer work in a cancer treatment? We mostly work on cancer treatment, as well as we have started working on the antimicrobial activities recently. So just to explain the process, like conventional drug molecule, there is a multi-drug resistance cancer cells. So you need to increase the dose if you have a resistance. And if you increase the dose, there is a toxicity to body. So our amphiphilic dendrimers can carry these conventional drugs or oligonucleotides or the imaging agent. And we can use them to treat the resistance. They facilitate entry of the drug and oligonucleotide through the EPR effect. That is called as enhanced permeation retention effect, because the cancer cells, they had a wide open cellular structure, and our dendrimer with the small nanoparticle size can enter in deep penetration. And with that, we can achieve the anti-cancer effect. And also, we can encapsulate the imaging agent, where we can see the progression of the disease also. So with the help of our amphiphilic dendrimers, we can use drug molecule, oligonucleotides, imaging agent, and we can fight against the cancer resistance. So first, I will start with the siRNA delivery with our amphiphilic dendrimers, how we have achieved. So as I explained, these are the simple dendrimers with the hydrophobic tail and hydrophilic head with the primary amine on the surface, which was the first dendrimer. Later, we have got the evolution of our dendrimer depending on the properties we desired. We have to just put them in a water. They form self-assembly. they have a positive surface charge on them around plus 30. Then it's very rapid and easy. They form a nanoparticle size around 10 nanometer, 10 to 12 nanometer. They can easily encapsulate siRNA through charge-charge interaction, and the particle size was around the 100 nanometer. We have shown that our amphiphilic dendrimer protects the siRNA from degradation as our conventional dendrimer. And later, we have achieved the gene silencing on mRNA as well as protein level in vivo. So these dendrimers serve the purpose, what we wanted, the siRNA delivery, protecting the siRNA, and deliver it to the targeted site with the very good in vivo activity, as well as we have achieved a very good easy synthesis pathway as compared to the conventional dendrimer. Later, we have modified our dendrimers later, like instead of primary amine, later we put arginine on the surface. As we all know, arginine binds to the cell membrane very easily as compared to the other functionalities, because it has interaction with the phosphate and sulfate and hydroxy group. It can form a divalent hydrogen bond. So you can see in the screen, like the arginine dendrimer has outperformed the amine dendrimer, and it has showed a better cellular uptake. So this way you can modify the dendrimer surface based on your desired effect. You can attach different functionalities, sugars, proteins, siRNA, metals, because it is easy for synthesis as compared to the conventional dendrimer. Later, we have also explored our dendrimers for a targeted SRN delivery. For particular diseases, you need to achieve some targets to have a better therapeutic effect. So, how our targeted SRN delivery system works? So, it's very simple. It's not like the other delivery system where you have to go through really complex processes. Our SRN delivery system is not at all complex. Again, you have to just put the amphiphilic dendrimer complex with the siRNA, and we attach the peptides, and the peptide has a negative charge surface on it for its state, and it is easily attached on the surface of the dendrimer system. Later, we have used the RGDK peptide here, which target the NPR1 and integrin receptor. In a cartoon, you can see it is easily penetrated, then there is endosomal escape, and the mRNA downregulation is there. We have used HSP27M mRNA as a model siRNA, and you can see that with the help of RGTK, we have achieved a significant effect as compared to the only siRNA AD. Our siRNA amphiphilic dendrimer complex already have shown better effect, but to make it more better, we use a targeting moiety, and targeting moiety has shown a significant effect of its targeting. So from this, we can say that our systems are also easy to have a targeted system, and we have successfully achieved to stop the tumor growth in a mice model. Later, apart from siRNA delivery, we work on the drug delivery system, mostly the hydrophobic drugs, because the amphiphilic drug from a mice cells, and they have a really wide hydrophobic core, and in which we can have a drug loading, hydrophobic drug loading. So just to compare with the lipid and polymeric nanomicelles, they have a dense core. So they have a difficulty achieving maximum drug loading. But in terms of dendrimer, you can see in a cartoon, we have achieved a really good core where we can have enough space for load more drug. And we are working since last 10 years on the drug delivery, and we have achieved almost all the drugs more than 40% drug loading, and the micellar size is between 10 to 20 nanometer, but the average was 11 nanometer. And this is one of the very successful dendrimer from our group, YTZ3315, and it has been used for a drug delivery system. We use doxorubicin as a model drug here. Doxorubicin, though it's a water soluble, we convert into the water in insoluble form by treating it. Later, our dendrimer can encapsulate hydrophobic drug. It has a small size. Then it can penetrate into the tumor with the EPR effect, as I mentioned, enhanced permeation retention effect. As the vascular nature of the tumor, it get increased in case of cancer, and you can easily have accumulation of dendrimer, which has a low particle size. And we have seen our dendrimers can penetrate deep into the tumor, which is really essential for a potent anti-cancer effect. And we have seen the tumor growth has been stopped or reduced completely in the animal model. Then we have checked the effectiveness in a different resistant cells. And we have seen that our dendrimer system can achieve overcome the drug resistance in a different cancerous cells. And when we wanted to study what is the pathway of our dendrimer based drug delivery system, and we have found out that the micropinocytosis is the pathway for entering our dendrimer drug delivery system into the cancerous cell. And again, there is endosomal escape, and there is a release of the drug to the nucleus where we want a desired effect. The third section of our work in our group we do is the imaging and the optical imaging. As I mentioned, imaging is a really essential part in a cancer treatment because we need to study the disease progression. So we wanted to explore the application of our dendrimers for the imaging purpose also. So how we do imaging? We do imaging by two ways. Okay, so we do the optical imaging where we encapsulate hydrophobic dye, and we do the radio imaging where we conjugate the radio ligand on the surface using some scaffolds like NOTA and DOTA, and then we can study the progression of the cancer progression. So I will go one by one for the optical and the radio imaging. And as I mentioned, the multivalency and nanosize improve the image quality as compared to the conventional marketed agents like FDG. So just to show how we significantly improve the near infrared imaging with the DIR dye. So here you can see the DIR is a hydrophobic. It is easily encapsulated. You have to just inject to the mice and it forms the stable nanoparticles. Here you can see the two graph. Here is the encapsulated in interior of supramolecular dendrimer and in dissolved in the MEOH, methanol. And you can see the intensity is similar. So there is no change in the intensity or the activity of the dye. And later, after injecting to the mice, we can see we have achieved a very good localization of our dendrimer nanosystem in the tumor starting from 2 hours to 48 hours. So we can easily image the tumors and we can easily study the disease progression using this imaging agent. The another aspect for our dendrimer nanomicelle system is for positron electron imaging, PET imaging, where we use a different scaffolds like DOTA and NOTA, and we use a different radioligands like gallium, gadolinium, indium, and the principle is the same. Here we attach them on the surface. Later they form a self-assembly which has a particle size in between 10 to 20 as I mentioned, and we have achieved a successful imaging of the tumor using the PET. Similarly, we have shown application of our dendrimer using the SPECT imaging where we have used the indium. And later recently we have published the another SPECT imaging supramolecular application using the indium, using the two different scaffold, DOTA and NOTA, which chelate the the radioligand, and we have successfully achieved the imaging contrast as better than the marketed imaging agent. So to conclude at the end, as I mentioned, the first problem what we had, like when we go for any research project or any problem, we had a problem of the drug delivery. For a conventional drug, we had a problem of their solubility, viability. For siRNA, they has a problem, or the nucleic acid therapeutic problem of degradation and reaching to the target site. So to overcome them, we work on a conventional dendrimers, which were better, but they had a problem in a clinical trials. So to solve that problem, we again rationally work on the structure of the dendrimer, and we successfully design a dendrimer which can be easily synthesized with less and less impurities and very high yield. So the GMP production can be achieved with the ease. And later, we have shown that after the GMP production of our amphiphilic dendrimer, like the qualities of GMP production, they have the similar capabilities or better capabilities than the conventional dendrimer, where they can encapsulate hydrophobic agent, hydrophobic drug for drug delivery. We can encapsulate nucleic acid therapeutics with the charge-charge interaction. We can do targeting, and we have also shown our dendrimer application in optical and radio imaging. And recently, our amphiphilic dendrimer has been scheduled for a clinical trial, and I think the clinical trial might start in 2021. We hope so. So I would like to acknowledge all my group members. This is a really old picture, but we have a lot of new students now, and different fundings has been achieved in our group through CNRS, Marseille University. There are different European projects are going on. Myself, I am working on the three projects currently. One which was completed was target for cancer. Then there is one for a brain infection, and another for the glioma, for the brain tumor targeting. And we have achieved our all results and success with our collaborators. I would like to thank our collaborator from different countries and the different institution. And thank you. If you have any questions, suggestions, please, and we welcome always the active collaboration. Thank you so much.
Thank you, Dr. Dinesh.
Hello.
Yes, Dr. Dinesh. Hello.
Yes, sir.
Yes, Dr. Dinesh. Yes, sir. Thank you. I would like to appeal delegates. If you have any question, please drop your questions in chat box so that I can ask your questions to Dr. Dinesh. Please. Yes. Any question from delegate? Just a minute.
Yeah, I'm waiting.
Yes, Dr. Dinesh, I have one question.
Yes, sir.
What about the manufacturing cost of these dendrimers as compared to other nanoparticles?
Okay. So first, I would like to explain, like to achieve a final dendrimer, we need at least 2 months to go from the start to the end in our lab, and depending on the desired dendrimer, it involves 9 to 16 different steps to go it, and depending on the type of material we use. But yes, if you have to say in terms of cost, they might be a bit costlier than the other polymeric and lipidic material for sure.
Yes.
They are more expensive in terms of their production.
Okay. There is one more question from Dr. Shilpa. Major characterization parameter for dendrimers.
Okay, it's a very good question, because it's always important to characterize your dendrimers in a very good manner. So when we have to do the characterization if I have to start from a synthesis, all the synthetic steps, like for just visual characterization, we go for IR and UV, but at the end, for the final products, we rely on proton NMR, then carbon NMR, then 2D NMRs, and we need a really high resolution mass spectroscopy. So in our group, like my boss is really strict. We need a really pure dendrimer, because the impurity effects, and to see the impurities, you need a really good techniques to analyze these dendrimers. So we mostly rely on a really high resolution NMR. Sometimes if we don't get a really good result with 400, 300 megahertz NMR, we go for sometimes 600 and 700 megahertz NMR also. As well as we like to do the impurity profiling using the high resolution mass spectroscopy, where we use LC-MS to analyze the impurities. So these are the major characterization parameters. And then for their physiochemical characterization, the first thing we check the solubility, whether it's soluble in water or not. Our most of dendrimers, what we work, depending on the surface, they are water soluble. If I have to tell you, approximately we can solubilize 40 to 60 milligram of our dendrimer within 1 ml of water. So they are very highly soluble. Next, after solubility, we check their particle size, their zeta potential, micelle concentration as they form a micelles. So we do their CMC using a different fluorescent technique, which are more reliable, using different dyes, or we use the ITC, which is isothermal colorimetric assays for a precise CMC. Our dendrimer has a CMC, critical micelle concentration, ranging for 4 micromolar concentration to 100 micromolar, depending on the generation, because currently we have a set of more than 70 different dendrimers, 70 dendrimers in our group, and they range from a different. But the one which are more successful, they have a CMC below 20 micromolar. So with a very less concentration we can achieve the micelles. And later we check their toxicity, which is another major characterization parameter, whether they are toxic and non-toxic, which is we check on the different cell lines as well as the cancer as well as the normal fibroblasts. So this way we process for the characterization of our dendrimer for a physiochemical and structural characterization, and once we achieve this, then we go for their application, biomedical application. But we never go for a biomedical application until we really confirm they are structurally pure and they have sufficient physiochemical properties to achieve our desired effect.
Okay, there is one more question, Dr. Dinesh,
Yes,
from
sir.
Dr. Nitin Jain. Till any product in market based on dendrimer for gene therapy?
No. Till now there is no problem.
Okay, okay. Thank you very much. Thank you.
But I would like to answer more. Like even the lipid particles research was going since last 20 years, and just last year there are 2 molecules have been approved by FDA. So like lipid has started 20 years ago, and after 20 years we have achieved something for gene therapeutics, and we are hoping for a dendrimer also. We started working since last 10 years, and there will be a clinical trial. We hope so that our product might be useful clinically also.
Okay, Dr. Dinesh, shall I ask one more question? The
Mhm.
question is uh, Dr. Vikas Dash. What about stability of dendrimers?
Okay, so stability of dendrimers depend on its core, which core you have. Like the dendrimer we are working, they are very stable. We always check their benchtop stability, their stability at 40 degrees Celsius, there's stability at -20. We usually store our all dendrimer if we don't use in -20, but we has its solution like in a water solution, and we have kept in the refrigerator normal 4 degrees Celsius, 7 days, 20 days, there was no problem. On a benchtop also we have seen, like we have checked for 3 to 4 days stability. There was no problem with our dendrimer. Even with the dendrimer encapsulate system, sorry, the drug encapsulate system of dendrimers have shown a very good robust stability on the bench surface. There was no drug release, or there is no precipitation we observe. So it is because we use the PAMAM core. But if you use the ester core, which is another purpose where you want easily degraded dendrimer. So for ester core, the stability is always like very less. Your dendrimers can degrade in 2 to 4 hours, depending on the core and the functionality you have. But in our case, our dendrimers are very much stable.
Okay, okay. Thank you, Dr. Dinesh.
Okay,
Dr.
sir.
Dinesh, you thrown a light on preparation of dendrimers and what exactly the research is going on in Dr. Peng's laboratory
Okay.
through Dr. Peng's team. I'm very
Mm-hmm.
much thankful to Dr. Dinesh. I would like to share one quote
Okay, sir.
about the presentation and about the efforts you're taking at France.
Okay, sir.
Knowledge is no value unless you put it into the practice. And this is the famous quote by famous Russian writer Anton Chekhov. So you brought the knowledge in the practice itself. So I'm thankful to Dr. Dinesh for his valuable guidance, his valuable session. I'm also thankful to Professor Hemant for moderatorship of this particular session. All delegates are requested to submit attendance and feedback. The link is already dropped in chat box. Please go through the link and submit the attendance and feedback immediately. Thank you very much. Thank you, Dr. Dinesh. I would like to say thanks to you on behalf of organizing committee members, my college principal, Dr. S.V. Amrutkar, sir, and whole team of Gokul Education Society, sir, Dr. M.S. Gosavi College of Pharmaceutical Education and Research. And I'm thankful to Dr. Dinesh on behalf of Indian Pharmaceutical Association Nasik branch, because this e-FDP is arranged in association with IPA Nasik branch. I'm thankful to Dr. Dinesh. So this is the end of the first session of the day 2. I request all the delegates be tuned at 3:30 sharp. There will be next session by Dr. Vivek Borse, who is DST Inspire faculty at IIT Guwahati. Thank you all the delegates. Thank you, Dr. Dinesh. Thank you very much.
Thank you so much, sir, for giving me an opportunity to talk. And even I would like to have the feedback from the delegates and the listener, because it will help me to improve my presentation and the sliding experience.
Definitely, definitely. I will share feedback in coming 3 or 4 days. Yes, yes.
Okay.
Thank you, Dr. Dinesh. Thank you.
Thank you so much, sir. Thank you. Bye-bye.
Thank you.